A very good afternoon to everyone. First of all, I would like to thank the organizers for giving me this opportunity. And I'm very thankful that I got a platform for sharing my views on the topic, which I'm going to share through my presentation. Moringa oleifera, the Pharmacogonosea. As we all know that after COVID, we have seen a shift of finding natural alternatives, whether it comes to therapeutic applications or for food preservatives, we are finding more options of natural origin, in which Moringa oleifera stands as a potential candidate because it all parts, starting from roots, leaves, seeds, each one has many medicinal properties. We are going to see how this ethnomedicinal legend is still relevant in the present scenario where we are discussing various diseases and their complications and why the only synthetic route is not sufficient to cure that disease. We have seen a large number of people are affected by especially the diabetes disease, and the number is increasing day by day, and the solution is still not, we can say, accurate, and we need more and more, we can see more and more alternatives. And when it comes to the natural part, Moringa oleifera and other plants like we can see Moringa oleifera has a potential applications. It is mentioned in Charak Samhita around 1000 BC. The traditional use of these Moringa oleifera decoctions are already mentioned, and in various Ashtang, Hridaya, and Yog Antakara, it is seen like seeds, oils are used for treating different earaches and inflammation. And the modern validation, when it comes to modern validation or its clinical reality, standardized extracts are entering into the randomized controlled trials for type 2 diabetes, neuropathic pain, and synthetic preservative replacement. When it comes to modern validation, identification of high amount of folic acid and glucomoringin, which has suppressing pro-inflammatory agents like cytokines and the potent antipyretic mechanisms and wound healing acceleration via vascular endothelial growth factor regulation is also got mentioned in various studies, which shows the modern validation of these ethnomedicine from the ancient times to the modern times. When it comes to the whole, when we see why this plant is so much have importance, so the leaves, the seeds, the roots, every part has its own importance. And the dominant components, like in leaves, we have quercetin, camphorol, having phenolic component, which has therapeutic actions like potent antioxidant, blood glucose regulation, down regulation of lipid peroxidation. And in seeds, we have glucomoringin and oleic acid, cardioprotective nature, anti-inflammatory nature, and water purification, having antimicrobial isothiocyanates also. When it comes to roots, it has dominant compounds like aurantiamide acetate and 1,3-dibenzylurea, which has a property of inhibiting the TNF alpha factors production, having potent analgesic and anti-ulcerogenic effects. So we can see their leaves, their seeds, each one has therapeutic applications. But how the problem arises when it comes to we see different types of pathological cascades related to different diseases. Here we are focusing on the, we can say, the diabetic part or hyperglycemic part. This figure shows the pathological cascade of hyperglycemia, which means that the excess glucose is present in the blood, and which leads to, uh, uh, we can say, increase in reactive oxygen species. And this generation leads to this generation of reactive oxygen species generation leads to the depletion of body's natural antioxidants such as super oxidase, dismutase, catalase, and other antioxidant enzymes. And this result in tissue damage, lipid peroxidation, and DNA damage, leading to organ failure like nephrotoxicity and hepatotoxicity. So here now the role of Moringa oleifera and how its mechanism play in this condition, especially when there is a reactive oxygen species generation and there's a depletion in antioxidant whole mechanism. So how this Moringa oleifera is going to work, how it's going to suppress this condition. This is one of the video where I'm showing like how these quercetin and camphorol provides the external defense, whereas the internal defense includes the aggressively moringa used to activate and provide the enzymatic boost to the whole process. So in this figure, we can clearly see like the external defense involves the direct free radical scavengers intercept oxidative agents before membrane penetration, like reactive oxygen species, TNF alpha factors, and interleukin 6 factor. And the internal defense from this Moringa oleifera involves activation, enzymatic boost, and outcome involves the reverse lipid peroxidation and halts the hyperglycemic induced organ damage. In activation process, Moringa aggressively upregulates the NRF2 signaling pathway and triggers massive intracellular production of superoxide dismutase, catalase, and glutathione. So this, the both mechanisms helps in the hyperglycemic condition, which we are discussing in the first slide. This we can see in this slide, there's how the threat, how the Moringa barricade involves and how this nuclear exclusion takes place. In the cytoplasm, there is an inflammatory stimuli attempt to phosphorylate different factors, and the Moringa bioactive compounds physically prevent the phosphorylation of particular or the degradation of the factor. Because or out of this action, transcription of inflammatory protein is halted, and it marked down regulation of COX-2, INOS, TNF factor, and interleukin 6. So when it comes to the preclinical efficacy of type 1 diabetes and type 2 diabetes, there are different models in which this has been applied. Like for type 1 diabetes in Alexion-induced mice and rats. In type 2, we have high-fat diet genetic models are used, and doses are also found like 100 to 300 mg per kg of leaf extract and 150 to 300 mg per kg of in case of type 2 diabetes. The outcomes in case of type 1 diabetes, it restores insulin secretion, drastically reduces renal hepatic damage, and accelerates diabetic wound healing and VEGF upregulation. Reverses hepatic liquid accumulation. Outcomes in case of type 2 diabetes, it involves reverses hepatic liquid accumulation, downregulates adipogenesis genes, and decreases leptin and improves overall insulin tissue sensitivity. So we can see two different key outcomes are found when it comes to type 1 diabetes and type 2 diabetes. There are studies that are proving that how this Moringa oleifera is a very potential candidate, and this is the clinical trial dossier, which is still needed to be translated into the human outcome. But still, like some of the studies are showing, like the Anthenon et al. In 2016 has reported 4-gram leaf powder capsule to the healthy subjects, and it shows significant increase in baseline insulin secretion following a single dose, whereas Dion et al., Dixit et al., and Gomez Martinez et al. Shows different outcomes like improved postprandial blood glucose response compared to non-diabetic controls, improved LDL HDL ratio, reduced waist hip circumferences, significant improvement in fasting blood glucose and glycated hemoglobin. So these are some positive results, but there are still more to be translated when it comes to the human trials or human outcomes. Uh, there are still, but there are not everything is good or everything is positive. There are some, we can say the red zone is also found and genotoxic effects are observed at supra supplementation level. And especially in case of pregnant ladies or it is found that it is not good for them. And as we can see, like in the greener zone, 100 to 300 mg per kg consistently shows high efficacy with no organ toxicity. The red zone involves the supra supplementation level, and the yellow zone where cautionary limits lethal dose in rat models exceeds 2000 mg per kg, and the leaf extracts exhibit abortifacient properties of high doses. Ingesting during pregnancy carries risk of severe uterine wall contraction. The second aspect, apart from the therapeutic or the diabetic part, they are found to be important against or as an important alternative for synthetic additives, because we have seen different carcinogenic effects and allergic reactions are we can say are found when it comes to using these synthetic preservatives. Some reports are also there, but we have not many options, because whether it is come to essential oils or any other alternatives or any other plant extracts, volatility, stability, oxidation is one of the major issue. That's why we are still reliant on the synthetic preservatives. So in case of moringa, uh, it has multi-target potent antimicrobial properties against Staphylococcus aureus and Aspergillus species, having high antioxidant polyphenolic content. They are completely non-toxic at standard preservation concentration. And when it comes to label impact, it is clean label, natural preservative, offering collateral nutritional benefits. So the phytopharmaceutical pipeline of this Moringa oleifera extract, nanomicelles, hydrocolloid films, effervescent tablets, thermoreversible in situ gels, all these are some of the, we can say, the pipelines where we can utilize this seed oil or leaf extract for the particular purpose. Like for hydrocolloid films, it accelerates cell proliferation in diabetic excluding moods. And delivery system, like effervescent tablets, thermoreversible in situ gel. So we are working on this for getting better results or for getting proper, we can say, the path from where we can properly utilize its potential for the particular purpose. The dossier, clinical data, botanical data, and commercial data. It is the master modulator of cellular defense. It executes one core chemical strategy with absolute precision. It aggressively upregulates endogenous antioxidant defenses, SOD, CAT, GSH, while physically barricading pro-inflammatory cascades like different factors. In diabetic kidney, this mechanism halts lipid peroxidation and tissue necrosis. In an excluding wound, its mechanism stops inflammation, allowing VEGF factors to trigger tissue repair. And in commercial food metrics, the same mechanisms halts oxidation and microbial degradation, vastly extending shelf life. So the benchmark, if we come to Moringa oleifera, we see the pharmacogenicity benchmark, scientifically validated decades of in vitro, in vivo, and clinical data confirm its efficacy as a potent hypoglycemic and anti-inflammatory agent. Commercial and clinical viability, from hydrocolloid colloid wound dressings to clean label Fera food preservatives, formulation engineering is solving the bioavailability issues. And the future, when it comes to its future, it represents the gold standard of modern pharmacognosy, where ancient ethnomedicine is perfectly mapped, molecularly verified, and technologically applied. So these are my some references, and thank you so much for patience.